Hello, students. This is Dr. Faizan Mirza. In this video, I am just covering with you the review of how the phospholipid bilayer looks like under a microscope in a transmission electron micrograph, and how the phospholipids look like, how the phospholipid tails look like. You need to just recall what we have discussed already, the cell surface membrane. So if this is the cell surface membrane, this is the inside of the cell, and this is the outside of the cell. If I see this particular black line in a transmission electron micrograph. If I use a transmission electron micrograph to view this, I'll see these three layers in one membrane. There will be a dark line and there will be another dark line, and sandwiched between them will be a lighter shade. The dark line at the top are the phospholipid heads. The dark line at the bottom are the another phospholipid heads. So they both are the two monolayers superimposed on one another, and the lighter region between are the hydrophilic tails. So this is how the membrane looks like. Now the question here deals with the UK. This question came in Feb March 2023, variant 22. So this came in, the table is given for eukaryotic and prokaryotic cells. You are given various cell structures, whether they are present or they are absent. First row is already done for you, and you are asked for Golgi body, circular DNA, ribosome. Tick for the eukaryotic cell, and cross for the prokaryotic in the Golgi body, where circular DNA and circular ribosome are present prokaryotic cells. So a tick there. It's a 2-mark question, so you know a 2-mark question will be marked in the form of rows, or sorry, in the mark of column. Both the column must have all the correct options stated by the student to get 1 mark for that particular column. So 1 mark for the eukaryotic column, 1 mark for the prokaryotic column. All the options must be marked correct. Now, for example, if the question was given 3 marks, if there were 3 marks, so they would be giving you marks for all rows. The rows would be marked, and but you need to just know how the marking is done to when you are attempting this kind of question. All the cells have two cell surface membranes. All cells have a cell surface membrane, not two. All cells have a cell surface membrane. Figure 1.1 shows transmission electron micrograph of a part of a cell of two adjacent animal cells, cell 1 and cell 2. So this is cell 1, this is cell 2. So this must be the cell membrane of cell 1. This must be the cell membrane of the cell 2, somehow like this. And we know it's a transmission electron micrograph. You will see a dark line and a light structure here, and a dark line and a light structure here, because both must be phospholipid. They have mentioned this as a box R. The next they are saying in the diagram, in the space below, draw a diagram. Drawing comes in the paper. Draw a diagram of region in the box labeled R. Your diagram should show 4 dark lines. The 4 dark lines are 1, 2, 3, and 4. All the 4 dark lines must be there. The label diagram to identify what is shown by the dark lines and each of the 3 spaces between them. So space for the diagram, you will draw 1 dark line and the other dark line. Space should be there, and then the dark line, the other dark line. The width, or you can say the width between the space between these 2 dark lines should almost be similar, because this is what's shown to you. The width here and the width here is common, and the width in between these two is much more. So I'll make a diagram which is more relative to the one which I have seen above. So phospholipid heads are represented by these dark lines. This gap in between these is the intercellular space, the space between the two cells. So this is what you can mention. Then this region here is the non-polar fatty acid tail. You can mention one, or you can label for the other as well. This is the again the non-polar fatty acid tail. So this will get you 3 marks. Furthermore, this question goes on to another chapter altogether. They're asking you for mitogens are short chain of amino acids that function as cell signaling molecules. Mitogens are released from the cell secretory cells, travel to the blood to the target cells, where the mitogen bind to cell surface receptors. They are cell signaling molecules, so they must bind to cell surface receptor. The target cells respond by progressing from G1 phase to S phase for the mitotic cell cycle. We know in G1 phase the growth occurs, and in S phase DNA replication occurs. Outline what happens in G1 phase and the S phase. G1 is for growth, RNA protein gets synthesized. S phase for DNA replication by semi-conservative replication. The DNA content become twice. So these can be your points for this question. As a result of mutation, the production of release of mitogens in the blood can be greatly increased. Suggests a possible consequence of target cell of increased concentration of mitogens. Since the mitogens are promoting the cell from actually affecting the G1 and the S phase, so if mitogens would promote are there, they would promote more cells who are in G1 stage to begin S phase as early as possible. So they will go to the S phase, and the DNA replication will occur very fast. Before the actual growth would have occurred, this may lead to tumor formation. So basically, I had to cover how the diagrams need to be drawn and how to interpret these diagrams where gray lines are shown to you. Most of the diagrams will come always from a grayscale image. You need to know how to interpret that and redraw or just label it accordingly. That's it from my side. I hope that was helpful for you. Thank you so much.